Okay, so we are going to talk about a catabolic process called glycolysis. So remember, catabolism is when we are taking a bigger product and breaking it down into smaller pieces. So the overview of glycolysis is that we start with 1 6-carbon glucose molecule, and throughout 10 steps, 1, 2, 3, 4, 5, we turn it into 2 3-carbon pyruvate molecules. So we have 1 6-carbon glucose, and we're going to break it apart into 2 3-carbon pyruvates. So you know this is catabolic because you're going from 1 bigger molecule to 2 smaller molecules. And glycolysis has 2 major phases. The first is called the energy investment phase. The energy investment phase, we are actually going to put in 2 ATP to ADP. We're actually going to have to invest some energy in. And then in the energy generation phase, we are actually going to make 4 ATP, for ADP, and then these are 2. We also are going to reduce 2 electron carriers. NAD plus is going to get reduced to NADH. So overall, the whole 10 steps of glycolysis, we put in 1 glucose, we're going to put in 2 ATP, we're going to get out 4 ATP. So net, what is happening is 1 glucose is producing 2 pyruvates. We are converting 2 ADP to ATP because we are using 2, making 4, so a net of 2. And we also are reducing 2 electron carriers, NAD plus to NADH. So this is a 10-step process. We usually call reactions in biochemistry pathways or cascades because it's most of the time not a one-step substrate to product. Usually there's multiple steps. And this allows for regulation, and we'll talk about that as we go along too. So I'm not going to draw out all of the steps of glycolysis. You can see all of these in your textbook and in your PowerPoint lecture. We're going to go through each of the steps of what's happening. You should go through each of the steps of glycolysis on your PowerPoint slide. What you should pay attention to are what we talked about when we talked about metabolism. You should pay attention to any steps where there's a change in the carbon structure. You should pay attention to the transfer of phosphoryl groups. This will usually involve ATP production. And then you should pay attention to electron transfer. So NAD plus or NADH. Okay, so the first 5 steps are called the preparatory phase or the energy investment phase. And each of these steps is catalyzed by an enzyme. And you'll see the enzymes listed on the slide or in your textbook. And then you'll also see the structure of what is happening after each of these chemical reactions. So essentially what's happening in the first step, we have glucose, we're adding a phosphate, so we go from glucose to glucose 6-phosphate. And that's catalyzed by the enzyme hexokinase. So hexo means 6, kinase means this is an enzyme that's transferring a phosphoryl group. That's what a kinase is. It's an enzyme that transfers a phosphoryl group. Next, we go from glucose 6-phosphate to fructose 6-phosphate. So we go from a ring that has 6 sides to a ring that has 5 sides. We're making this molecule more symmetrical so that when we split it, we're going to get 2 identical molecules eventually, because remember, we're going to end up with 2 pyruvate molecules. We also are going to add another phosphate. We're going to add another phosphoryl group, so transfer of a phosphoryl group. So now we have fructose 1,6-bisphosphate. So we have 2 phosphate groups, one on the first carbon, one on the 6th carbon. Fructose is a 5-carbon ring, heterocyclic, meaning there's an oxygen in that ring too. And then the next step is catalyzed by an enzyme called an aldolase. And what an aldolase does is it splits this ring and it produces 2 3-carbon molecules, a glyceraldehyde 3-phosphate and then a dihydroxyacetone phosphate. The glyceraldehyde 3-phosphate is ready to go into the next step here. The dihydroxyacetone phosphate needs to be converted into glyceraldehyde 3-phosphate. So that is what the triose phosphate isomerase does. So now from this point on, right here, we're starting with 2 molecules of the glyceraldehyde 3-phosphate. This is on slides 28 and 29. And then you'll notice on the top of 29 where it lists all of the names of these intermediates, you'll see a 2 in parentheses after it. That means you have 2 of each of those molecules because we split that 1,3-bisphosphate. Okay, so then we are going to reduce the electron carriers and oxidize and phosphorylate the substrates. And we're going to make 2 ATP by removing the phosphoryl groups off the 1,3-bisphosphoglycerate. And then a few more steps, we are going to form 2 molecules of pyruvate. So these are the 10 steps of glycolysis. After this, I have a slide that goes through each reaction in detail. One thing that I want you to pay attention to is that anytime you have an enzyme that is involving the transfer of a phosphoryl group, anything that's making ATP or ADP, you're going to need a cofactor of magnesium. Magnesium, this positively charged cation, is going to stabilize the negatively charged phosphoryl groups. So that's the purpose and why you're going to see the magnesium ion above the arrow for these reactions. Any reactions that are involving the transfer of a phosphoryl group are going to require the stabilization by this cofactor. So the next few slides are going to go through step by step of glycolysis, what we just talked about. And let's see here. So again, I just kind of want you to understand the big picture that the first half is energy consuming. It's an energy investment. And the second half is energy producing, energy generating. So the second half of glycolysis, all of the reactions are also written out. Here on slide number 46 is a picture showing this magnesium ion stabilizing those negative phosphoryl groups in the transfer of a phosphate. So that's an important thing to look out for. Also, in one of these steps, one of the intermediates that we make is 1,3-bisphosphoglycerate. That's 1,3-BPG. So remember back to hemoglobin. Hemoglobin is regulated by 2,3-BPG. So remember I said that this was going to come back in glycolysis, so here it is. So here there's an enzyme that can catalyze a mutase, which switches this phosphate group from the 1 position to the 2 position. So now we have 2,3-bisphosphoglycerate. So there is, this is on slide number 50, and there is some oxygen saturation curves, and it shows a hexokinase deficient cell and a pyruvate kinase deficient cell, and then it shows normal erythrocytes in the middle. So that would be normal hemoglobin in the middle, and then it shows hemoglobin that's hexokinase deficient and hemoglobin that is pyruvate kinase deficient. This will be a test question. I am not going to answer it on the lecture because this is going to be a test question. So I want you to think about this. What explains these curves? And why would the oxygen saturation change with the deficiency of these 2 enzymes? So this is going to be on the test. Slide number 50. You will see that exact slide on your exam. Okay, so we get all the way down to pyruvate, and this is an overall energetically favorable process. The overall free energy of this is about negative 102.9 kilojoules. That's the free energy. So this is a spontaneous reaction, but even though we're investing some energy here, we're producing more later. So definitely read your textbook on glycolysis. I will ask you about some of the specific steps of glycolysis. Pay attention to where the carbon structure changes, transfers of phosphoryl groups. Anytime you're seeing electron carriers being oxidized or reduced, because we are going to see these reduced electron carriers go to what's called the electron transport chain. We're going to see this in a few more lectures. So for each molecule of glucose, we're going to see that we can make about 32 ATP. Here in this first step of glycolysis, this first set of reactions, we're only making 2 ATP. So obviously in the rest of cellular respiration, we have a lot more ATP to make. And the place where we're going to make most of the ATP is in the electron transport chain. And that's going to come from all of these electron carriers that we have loaded up throughout the glycolysis. And what's coming next is the this TCA cycle. So work through those slides on glycolysis. Make sure you can understand what's happening in each of these 10 steps of glycolysis. And also think about what's happening on slide 50, because you will see that on your exam.